get started. I'm Dixie Summers. I'm on the VSO board and want to welcome all of you for this evening's presentations. And happy to have you here. A couple of little procedural things first. We had asked that during the presentations, if you would keep yourself muted so we don't have any TVs in the background or things like that. And if you want to turn your video off, that would be probably good for your connectivity and might be a little less distracting for those of us watching. We're also going to use the chat for questions and answers. So if you have a question, please put it in the chat, which you can find at the bottom of your screen. If you put your mouse over, you'll see a little thing that says chat. And at the end of each presentation, Leslie will look at those questions in the chat and pose them to the presenter, so have a chance to get some answers. So with that, let me introduce Diane Kolowski. She's the VSO board president this year and has a few words to say.
Greetings, everyone, and thank you so much for joining us. We are really excited to finally be figuring out how we can offer these presentations. Typically, historically, they have occurred at our annual meeting in May in person, which, as you know, there have been no meetings in person since, since COVID. So Leslie, now these are our conservation grants, one conservation grant today, and award recipients that will be treated to their presentations on their research work that they did. A huge thank you, not just for joining us today, but those of you who have been supporting the VSO. This is one of the most important things that we do, and your donations and your membership contributes to this important work. Ornithology, in studying different topics of interest and hopefully getting some young ornithologists supported in the work that they would like to do. So without any further ado, a couple of things. Patty Rhyne has managed the conservation grants. She's a longtime enthusiastic board member that just this past year her term was up, and she's now off the board, but she'll be introducing the conservation grant recipient and his presentation. And then a huge thanks to Dr. Leslie Bullock, who has done just phenomenal work with the VSO, with managing the Murray Awards. And she will introduce the three recipients of the Murray Awards and their presentations. Leslie is a professor, Dr. Leslie Bullock, at Virginia Commonwealth University, and we're really blessed to have her on board. She also does extensive work with Bramble Hill. And I'll mention Patty as well established was one of the people that established the Kestrel Fund and the Kestrel Project, starting in Highland County. So with that, Patty, if you will take the wheel.
Okay.
Thanks again, everyone.
Thank you, Diane. I'm Patty Rhyne. The conservation grant was started in 2007, and every year the VSO awards these grants to worthy candidates that are conducting research in the field of ornithology or in conservation. And it's usually to benefit Virginia species and habitats. Grants are usually in the amount of $500 to $1,000 and sometimes more. We budget about $2,500 annually, sometimes a little bit more than that. The Conservation Committee reviews each application and decides who will receive a grant and the amount to be awarded. The committee then reports the selections to the full board. I've chaired this committee since 2007, and this year I'm passing my twirling baton to Dan Albert Mallinger, the new board member. After a project is funded, the researcher is asked to present their findings to all the members of the VSO. And as Diane has said, we usually do this at the annual meeting, but tonight we have this special Zoom presentation. So, Dr. Joe Kolowski, I was lucky enough to visit him. I guess it was just last year, and we were doing kestrel stuff together. He comes from the Smithsonian Conservation Biology Institute. He's going to present research that's being done in collaboration with the Clifton Institute on movement tracking and nest box surveillance. That sounds kind of spooky, doesn't it? Surveillance of the American kestrel. The VSO has funded this project for 3 years now, since 2021. We recognize how important this research is in identifying reasons for the decline of this species. So, Joe, take it away.
Okay, fantastic. Thanks a lot, Patty. Hello, everybody. Thanks for joining, and thanks, of course, to the VSO for the generous support of our kestrel project. Let me share my screen. Okay, I get a thumbs up, Leslie, maybe. Does that look good? Okay, great. Good. Well, I'm really excited to give everyone an update on our research, which we started in 2021 with support from VSO. Just to give a little bit of context for the project, one of the things that motivated us was the long-term declines in kestrel populations in North America that have been evidenced by breeding bird survey routes, like the data shown here for the Piedmont region, where our project is conducted, and also from migration counts and Christmas bird count data. But the other thing motivating us on the project is the strong level of interest in a lot of regional NGOs and agencies in promoting a more sustainable, biodiversity-focused management approach to agricultural lands that are obviously home to a whole other suite of grassland bird species also in decline. Many of the proposed actions or management approaches that these groups are discussing, like I'm showing here, will involve changes to the structure of these open habitats that the kestrel relies on. And so what we set out to do was to learn what habitats are selected by kestrels throughout the year and how that selection might change in response to different management approaches. So if you look at the list of possible reasons for the decline in kestrel populations, there are a number of them. I'm highlighting some of the major ones here. And you can see that habitat loss and degradation, clean farming, these are some of the main things that have come up. And that's primarily what we've focused on in our project. But we've also collected data on insecticide and rodenticide exposure and fledgling survival. So I'm happy to answer questions about that at the end, but I won't be focusing on that. So we set out to answer a number of what we felt were important research questions in Virginia. How much space do American kestrels use and rely on in the breeding season? How does this change between males and females and at different stages of the breeding season? What are the open habitats in this region that are preferred by kestrels? And again, how does this change throughout harvest cycles and seasonal growth? And then finally, how does habitat quality influence the movement distances or the parental behavior of breeding adults? So let's dig into it here. So our team monitors a network of about 170 nest boxes in Fauquier and Rappahannock counties, which you can see here. And starting in 2021, with support from VSO and other generous donors, we started capturing adults in nest boxes in both of these counties and attaching small solar-powered GPS units with the backpack harness, which you can see on one of our females here. So these units have just recently become light enough to track this species. This is the first study to track American kestrels throughout its range with GPS technology. And we're able to record a location every 20 minutes during daylight hours for these birds. The data is stored on board, and then we download that data with an antenna unit. So the units have worked quite well for us. I'm showing here a graph of the tracking duration of the 61 different kestrels now that we have attached GPS units to. And you can see that in 2022, we started attaching units to males. Those are shown in blue. And these are ordered by deployment date. So you can also see on the right side of the graph that in 2023, this season, we attached a large number of units. 42 new units were put out on birds just this past season. And so, on average, we've recorded over 1,700 locations per bird. And we've tracked some birds now for 3 consecutive breeding seasons. So we have now an enormous tracking data set that we're starting to work through to answer some of these questions. So early on, we realized that this tracking data was really unique and that we'd be able to fill some gaps in what's known about the movement ecology, basic movement ecology of the species. So just this month, we have our first paper coming out from the project in Journal of Raptor Research. And just to summarize some of the main points here, we document very small breeding territories in the species, about 78 to 80 acres in size, with very little overlap in neighboring territories. You can see a map of neighboring kestrel territories there. We also show the first description of extraterritorial forays. These are exploratory flights outside of the home range, and sometimes very, very long flights up to 128 kilometers during the nesting season. And we also document shifts in some birds to post-breeding home ranges, so abandoning their nesting territory and setting up new home ranges up to 12 kilometers away from the nest box. So just to show you what this might look like, this is Peaches here. We've been tracking her for 3 years now. And you can see her tracking locations in purple during the nestling stage. And there's the other active nest boxes in the white targets there. And then if we move to the fledgling stage, you can see that she essentially became semi-nomadic. We could not construct a home range for the bird because she was moving to so many different locations. But then ultimately in the post-breeding period in July, she established a completely new separate home range that you can see in red there in her red locations. This was 3.5 kilometers away from her nest box. So this is the first time this has been described for the species, and we showed this in about half of our females from 2021. The other thing we document in the paper is these foray flights, and I'm showing an animation of one of our females that ultimately took a number of long foray flights. You can see all of our monitored nest boxes in blue there. So that's a large study area. And you can see that in the post-breeding stage here, over the course of a couple days, she took enormous exploratory flights as north as Front Royal and all the way south to Madison. So these were huge flights, and eventually she came right back to her nesting territory and settled just about 1 kilometer away from her nest box. So we're really interested in these foray flights. We dug into this in the paper, but the big finding was that the rate of forays that the birds were taking depended on the time of the season. Most of the forays happened during the nestling phase. But really interestingly, the highest foray rate was for the birds that ultimately abandoned their nesting territories. And so this, again, happened mostly early in the season. And so it seemed like these birds were able to tell that their nesting territory was not sufficient to sustain them for the whole season, and they began taking these flights. And up to 60% of these exploration flights ultimately were to the territory that they ultimately changed to. So definitely, there's a clear sign that they were exploring for their post-breeding shifted territory then. So just as an aside, we also are able to collect some really cool data, having tracked this bird for 3 years. And I just wanted to show you some really exciting data that we got this spring. This is by far the highest resolution eastern kestrel migration route. Peaches, this bird was collecting points every 20 minutes for her southbound and northbound migration this past year. And she migrated to Georgia, onto a farm in Georgia. And in fact, this is the second winter that she flew to that same exact farm in Georgia. So we're really excited to get more data like this in the coming spring when a lot of our birds hopefully return after migration. All right, so the primary focus again here is the habitat use. You can see locations from one of our females here in 3 different stages of the season that we're focusing on, and it's overlaid on the different field types that we have in our region. So I'm going to show some some of the first stages of analysis here. This is results of a compositional habitat analysis. And I'll just walk you through this. Basically, what we're doing is comparing the habitat that the tracking locations are in to the habitat that's available to the bird in their home range. And so if you have a selection ratio, each point here is just an individual bird. And if you have a selection ratio above this dotted line, that means that you're selecting for that habitat. You're essentially preferring that habitat. And so here we have different habitats in different colors. And what I want to highlight here in the nesting phase, when we have birds in the box, we can see that the meadow, sort of native meadow natural habitat, is preferred by many different birds. You can see there's a big range across the different bird individuals. But on average, which we can see here, there is a preference for this meadow habitat. And hay is somewhat avoided. Pasture is used as it's available. And of course, we would expect forest to be avoided, which we can see here in most birds. But when we shift to the post-breeding time of the year, this is when vegetation is higher. This is when birds are not connected as closely to their nest box. We can see that meadow is actually avoided at this time of year. And we see preference in pretty much every bird that was tracked, strong preference for pasture habitat. So to us, this is really interesting. It brings up issues of whether the habitat around the nest box is what the birds are using all year, but also the fact that some habitats might be only sufficient to support the birds in only part of the year. So again, strongest selection for this meadow grassland habitat, again, that is really probably favored by most grassland nesting birds. Use of this habitat early in the season, but avoidance of it later in the season, shifting to pasture. And this has been confirmed by more complicated modeling analysis that we've just concluded as well. So the next phase of this habitat analysis is to bring in measurements that our team has taken of the height and the density of vegetation in all the fields that the birds had access to. So this was a huge effort. And so now we're bringing in this data into the modeling to try to distinguish between whether the birds are selecting for particular habitat types or whether they're really just keying into vegetation height or density. And that's what's going to allow us to connect our results to some of these management practices that some of these local NGOs and agencies are looking into. So in terms of what's coming next, we have a huge amount of data here. Obviously, just in the last year, we've started to be able to compare males and females, which you're seeing some examples of here in these figures with males in blue. And what we're hoping to ultimately do is bring in the habitat, bring in the seasonal differences, and finally bring in differences between males and females to really comprehensively describe what influences kestrel movements in this landscape. So we're really excited that Kaylin Wolfer, one of our longtime staff members, has just started a master's position at Virginia Tech with a colleague of mine, and she's going to be digging into all this information and trying to pull it all together. The other big thing that we added this past year with support from VSO was we established a huge network of video monitoring nest boxes. So I'm showing one of the boxes here. These are custom designed boxes that have special houses on top to cover this motion activated security camera, which is powered by a solar panel here. And so we had a total of 47 of these boxes used by kestrels. And so we have an enormous amount of video data to start going through and really opens up a lot of exciting new research questions for us. I'm going to show you some examples here. One of the things we're digging into is what are the species that they're bringing into the boxes to feed nestlings, and what's the frequency that they're bringing in those prey. So this is a skink that's being brought in by one of our tagged females. This is what looks like a nestling or a fledgling bird that's being brought into the box to, actually, this is the same female. And then this is a tiger swallowtail that's being brought into a nest, obviously an older nest. For some reason, they let the little guy have this one. I'm not sure why. And then we're also, obviously, because this is a declining species, we're interested in causes of clutch reduction and nest loss. This is a black rat snake taking a nestling inside a box. The second nestling here ultimately did survive. And maybe this isn't too surprising that black rat snakes are getting into boxes, but we have had some other surprises in the box. This is a red-bellied woodpecker that ultimately stole a nest out of the nest box. And we've documented this happening now from red-bellieds and from flickers. And so woodpeckers seem to be sort of an unexpected predator of these kestrel nests. So we're excited to collect a whole other season of data from our cameras to really dig into what are the threats to these nests in this region. And then finally, what I'm most excited about is connecting the movement of the adults with what's happening in the nest box. So every one of our tagged pairs this year was in a camera monitored box. So we know exactly where these adults are moving, and we know what they're bringing back to the box and how often they're doing it. So I'm really excited to try to figure out why, or what's happening in the nest box, for example, from a pair like this, which used a very, very tiny home range for the whole duration of the breeding season, compared to another pair like this in Rappahannock County, which used an enormous home range. Are they bringing back fewer prey? Are they bringing back prey less frequently? How is that impacting the success of the nest? These are all things that we're going to be able to tease apart. So quickly, just in summary, we've learned some things about how meadows are utilized. They don't appear to be sufficient to support birds for the whole season. And so that brings in some opportunities to potentially manage pasture for both kestrels and other grassland birds. We've got a huge tracking data set to dig into, and I'm excited to start to bring in the camera data that we're just now getting in to tie all that movement data with parental care, prey selection, and nest success. And then finally, we're excited to get a whole other year of camera data to dig into in terms of nest predators and what's happening inside these boxes. So if you go to the Clifton Institute website and check out their research page, you can actually check out an app that we've built that allows you to track the birds and see where all of our different birds are going. It's a really fun thing to dig into. And I want to just close by pointing out my two co-PIs on the project, Alan Williams and Bert Harris, who have been a huge part of the project and great partners in this effort since 2021. And also thanking the whole team. We had a big team here in 2023 to make this all happen. So I'll take questions now from the group, hopefully.
Amazing, Joe. That was a really great talk. Very
Excellent.
exciting stuff going on. All right, so we probably have time for just a couple of questions before we should probably move on to the next speaker. Oop, the chat just disappeared. Hold on, where to go? So there's a couple of questions here. What is the average distance that a young kestrel would go from their original home range?
A young kestrel, yeah, I'm not sure if you're talking about like first-year birds or what, but we have not. So we had VHF transmitters on fledglings for one of the years of the study. This was last year. I didn't talk about that. And the furthest that we documented those birds going was about 25 kilometers away from the nest box, but they were found 25 kilometers away the next breeding season. Within that first year, I think it was about 12 or 15 kilometers that we tracked a bird leaving from their natal territory.
And then Isaac asks if you have any insight, and I think you mentioned that this is the first time this kind of data have been collected, so you might not know this, but he's asking if you have any insight on how foray range might compare to Western populations.
Right, so this is the first study, like you said, yeah, first study to have GPS tags on kestrels. And so I'm really excited, these tags are now working well, and I think other studies are starting to use them. I know Florida study is putting them on, for example. So I'm really excited to see if what we're seeing is going to be replicated in other areas. Yeah.
Okay. All right, let's do one more question. Dan asks, have you observed differences in habitat use during the post-fledging period for adults with and without fledglings?
That's a good question. Habitat use with and without fledgling. So in the post-fledgling phase, they're essentially not tied to the nest box anymore. And so I'm not sure we would expect them to behave differently, but we will be digging into sort of what the habitat use is like during that fledgling phase and comparing that to birds that don't have fledglings. So that we haven't done that in detail, but I'd be interested to look at that. We haven't gotten into that level of detail yet. But yeah, because some of those birds are having to come back to the box, obviously, whereas the other birds are released from their connection to the box and can kind of go wherever they want.
Right. All right, selfishly.
Oh, sorry.
I was going to say, selfishly, I do have one more question.
Okay.
I'm curious. So I wasn't actually aware that kestrels were migratory. So I was really surprised about your result. And so are all individuals migratory? And if it's not all individuals, do we know why some migrate, or what's the difference between those that migrate and those that don't?
Yeah, that's a great question. So I had to cut that from the talk, but so far about half of our birds have appeared to migrate. We've only gotten migration data from 2 birds, but about half the birds disappear around migration time, and the other half stay for the whole winter.
And is it male, female, or?
So we've just started to get more males now. So I think we'll have a lot more information in spring when we see what all the data looks like and who comes back. And we don't have enough data to say like why some of the birds stay and why some of them leave. There is some data from out west from a new paper that tried to ask those questions. But one of the cool things they found was that it's not consistent in individuals. So they'll have some birds that migrate one year and then don't migrate the next year. And so there's probably a lot of factors at play. But yeah, we don't know enough in the East yet about that.
Awesome. It's amazing what we're learning from tracking individuals. Yeah. Great. Well, thank you, Joe. We're all clapping for you in our virtual way. But I appreciate your talk. This is really great. And the work that you're doing.
Thank you very much. Thanks to VSO.
All right. Well, now we're going to shift gears to our 3 speakers that received Murray Awards. And the first of which is Casey McLaughlin. So Casey, you can go ahead and pull as I'm doing your introduction, you can go ahead and share your screen. Casey received a Murray Award in 2019 for her research on stress and mercury exposure as a master's student at the College of William and Mary, where she worked with Dan Crystal. She is now working on a PhD in biological sciences at Virginia Tech in the Kendall Sewell Lab. So we're really looking forward to hearing your master's research.
Awesome, thank you. Can you hear me all right? Or?
Yes.
Okay, awesome. Well, thanks for having me. Yeah, I am presenting on my talk entitled Mercury Stress in Songbirds. Let's see if I can get this to go forward. There we go. And as you already mentioned, this was part of my master's thesis in biology at William & Mary. I worked with Dan Crystal, Eric Bradley, and John Swaddle on this project and completed my degree in 2021. Also on Zoom. It was still a pandemic at that point. And this was funded in part by the Virginia Society of Ornithology, and I'm really grateful for your contribution to this work. So what I'm going to talk about today actually is part of a larger project that I worked on as part of my thesis, which was entitled Mercury Exposure and Chronic Food Stress Elevate Corticosterone in Blood but Not Feathers. That's a bit of a mouthful. You'll probably, hopefully, understand most of it by the end of this talk, but I won't be touching on the aspect relating to feathers. If you have questions on that at the end, I'm happy to answer them. And rather exciting, this is very close to being published in a scientific journal too, so I'm pretty excited about that. So to kick this off, I wanted to first introduce the contaminant that I have focused on in my work, which was mercury. I'll represent mercury with its chemical symbol Hg throughout this talk for simplicity. But mercury you may have heard of in the context of not eating too much fish, for example. Mercury is a metal, and it's a really widespread and very persistent pollutant. Mercury is actually naturally occurring, and it's typically released rather slowly over time through things like volcanic activity and forest fires. But of course, humans have greatly accelerated its release into the environment through things like the burning of fossil fuels, its use in gold mining, and a whole bunch of other industries also make use of mercury. The end result being that we have a lot more of it in the environment over a shorter time span than we would expect over the natural scale. And this mercury, once it is emitted into either the air, the land, or the water, most of it ends up actually in bodies of water. So things like lakes, rivers, streams, and even the ocean. And whatever form of mercury is dumped into the environment, once it is in these bodies of water, there are bacteria in the water that convert that mercury into something called methylmercury. So this is an organic form of mercury. And in this context, organic is not the healthy stuff you're getting at the store. Organic is actually pretty bad. Methylmercury is what we would call very bioavailable. So it's very easily gets into living things because it looks like a lot of compounds that humans and animals have in their body. And it's not good for us because we don't need mercury. At these contaminated bodies of water, what ends up happening is you have little fish ingesting this mercury from the sediment or the water column, and then they get mercury in their bodies. These fish then get eaten by larger fish that end up with even more mercury in their bodies because they've eaten a bunch of tiny contaminated fish. Finally, at the top of this food chain are species like herons and kingfishers that are eating a bunch of these large fish and ending up with the most amount of mercury in their bodies. And this is a process called bioaccumulation, where a contaminant ends up at the highest concentration at the top of the food chain, basically. And we actually understand this process fairly well, and we understand a lot about how mercury impacts fish-eating species like herons and kingfishers. However, in more recent years, we've learned that songbirds at these contaminated sites are also impacted by mercury pollution. This is interesting because obviously I don't know too many songbirds that are eating fish, so we're not entirely sure how they're getting this mercury contamination. That's still an area of study. But we also don't know a lot about how mercury affects these birds because we didn't know that it was affecting them for a while. But this has been shown across a wide variety of species. And a lot of this work actually came out of the spill at the DuPont plant in near Waynesboro, Virginia at the South River site. So that's where a lot of the early work was done, but we're still trying to understand how this mercury pollution is impacting these birds. And so oftentimes when you hear pollution and birds, you might picture something like this, right? Birds dropping dead out of the sky, and this, sort of similar to Silent Spring and the DDT pollution. But that's not what we're seeing with mercury contamination in songbirds, and it's actually not what we see with a lot of modern day contaminants. Instead, what we see is that these birds are living with chronic, so long-term, non-lethal levels of mercury just in their bodies for a long period of time. And while they're not dropping dead from mercury poisoning, we know that mercury is not necessary for any function in the body, so this is not good for them. And we know a little bit about how mercury pollution affects songbirds in this way. For example, it's been shown that songbirds exposed to mercury have lower body mass, they have problems with immune function, and things like that. However, what I was particularly interested in my master's work was understanding whether methylmercury pollution in this chronic, non-lethal fashion disrupts something called stress hormones in these birds. And stress hormones are pretty interesting because they're environmentally sensitive, so they can change in response to changes in the environment, but they also affect a lot of different functions of the body, including some of the ones on the screen here. So measuring these stress hormones could be a useful, what we would call, biomarker of the negative effects of mercury exposure. That was the idea. But what do I mean when I say stress hormone? This is a really loose definition for a hormone in birds called corticosterone. This is very similar to the hormone in people known as cortisol, which you might have heard of or be familiar with. And essentially, this hormone has a lot of different roles. So at rest, there is a small amount of corticosterone just circulating in the blood at sort of baseline levels. And this has a couple different functions, but basically it's keeping that animal prepared to respond to a stressful event. And when there is a stressor, like a predator or something, walking along and scaring a bird, this basically kicks on the system called the hypothalamic pituitary adrenal axis, or HPA axis. And the end result of all these organs talking to each other is the production of a lot of corticosterone that then ends up in the bloodstream. And this does a couple different things. So this increased corticosterone promotes things essential for survival, so that fight or flight stress response. But it also inhibits things that are maybe not as necessary for immediate survival of danger. So things like growth and immune function and reproduction, which are important in the long term, but not if you're about to be eaten by, a hawk or something. And corticosterone, when it reaches a really high level in circulation, also can inhibit its own production. So it'll actually cause the animal to secrete less corticosterone to get back to that resting level. And this is important because really high levels of corticosterone sustained over a long period of time can have really bad impacts on health because it is constantly promoting that fight or flight response and reducing things like growth. Those are pretty important over the long term. This is like if you've ever experienced the phenomenon of you're really stressed studying for an exam and you push for like a week and then right after the exam you get sick. That's sort of how you can think of this effect occurring in animals, essentially. But how does mercury fit into this picture? Well, mercury might be working at any one of the points in this system. However, the end result, if it is indeed disrupting this HPA axis, should be a shift in this hormone at the end here, corticosterone, right? And if mercury is impacting the secretion of corticosterone, especially those resting levels, which give us a sense for an animal's preparedness for stress, that has implications for these other really important functions of the animal, right? And so that could be kind of bad if these are getting out of balance. So building off of this, the primary aim of this part of my research was to determine the effect of these chronic non-lethal levels of mercury exposure, like we see at, for example, the South River site, on the stress hormone corticosterone in songbirds. And because not a lot of work has been done on this, I wanted to do this in a controlled laboratory environment where I could really determine cause and effect. So, but I also wanted to make it somewhat realistic, right? So I both looked at mercury exposure on its own, and then I also combined it with a food stressor that was meant to mimic some of the stressful food conditions that birds might experience in the wild. And my prediction here was that if mercury is disrupting the HPA axis, then birds exposed to mercury should have higher levels of that stress hormone, corticosterone, at rest. So you can think of this as the birds being under chronic stress in very human terms. So to do this work, I used a model songbird called the zebrafinch. These guys are not native to the US, but they're really well studied and easy to breed in captivity. And they're a charismatic species. And what I ended up doing, I had 4 different treatment groups. So this was sort of a 2x2 factorial design. I had 2 groups that were not exposed to mercury. So that was the control group, which basically received no manipulation at all. And then I had a group that was the food stress group. So these guys just got that food stressor that I was talking about. And this consisted of removing the food source for a period of 4 hours a day at an unpredictable time on a short daytime schedule. So essentially mimicking, inconsistent food availability during winter. And this is a paradigm that's been used before by other labs. And then I have my 2 mercury exposed groups. So the first was just the mercury stress group. These birds only received mercury exposure at a level similar to what we would see in the wild. And that was basically just mixed into their diet. And then finally, the compound stress group was the group of birds that received both the mercury exposure and the food stress. Excuse me. But what did I actually see? How did mercury exposure affect the stress levels of these model songbirds, the zebrafinches? Well, this was the main graph from this portion of my research. And you can see on the x-axis, we have the treatment groups that I just showed you. So on the left is our control, and then on the right is sort of the different manipulations. And then on the y-axis is baseline levels of corticosterone. So that's essentially stress hormone levels at rest. So how prepared are you for a stressful event? If you're higher on this axis, you're a bit more ready to go and under a bit more chronic stress than if you're lower down. And as you probably already noticed, the control is pretty low compared to some of these other groups. So you might take a look at these 2 red groups here, the mercury stress and the compound stress group. Both had higher levels of corticosterone compared to those other groups. And so we can think of these birds as being more stressed with this exposure to mercury than birds that were not exposed to mercury. And there is a slight, you can see that the compound stress group is a bit higher than the mercury alone, but this wasn't actually statistically significant. We would need to do a little bit more research to actually see if this is a real effect. So, what are the main takeaways here? Well, mercury exposure in this system did indeed result in higher levels of baseline corticosterone in the blood, which essentially means that these birds had more stress hormone at rest. And this might be bad for individual health and fitness because of all those different functions that corticosterone can affect. However, this also suggests that corticosterone might be a useful biomarker for detecting negative effects due to mercury exposure. We do need to do a bit more research to really drill down on that, but it's an interesting possibility. But why do we actually care about this, right? Like, why even study this? You know, the birds aren't dying of mercury poisoning. Why do we need to know what this is doing? Well, as I'm sure you all have heard, over the past 50 years, there's been a decline of nearly 30% in bird populations across North America. And one of the most heavily impacted groups was songbirds. And this is alarming, to say the least, but it's also kind of interesting, and it tells us some different things. First off, we should definitely continue studying songbirds. Second, songbirds, that's a lot of different species of birds. And as you can see, these declines occurred across a really wide geographic area. And this, to me, suggests that there's not one smoking gun, right? This isn't the DDT killing eagles and robins, but rather it's probably a combination of multiple chronic, non-lethal factors chipping away at populations through their impacts on individual health. So it's not things that are necessarily causing birds to drop dead, but there are these multiple factors combining to slowly chip away at populations. And this includes things like habitat loss and climate change, but also pollution. And unfortunately, pollution, especially things like mercury, they're not really going away. It's really difficult to remove these things from the environment. And so it's really important to understand the sort of longer term impacts of lower levels of contaminants on bird populations. And so building off of this, I've sort of continued to build from where I started at William & Mary, and I'm now looking at how long-term exposure to lower levels of lead impacts songbirds as well at Virginia Tech. So here I'm interested in looking a bit more at developmental trajectories. So at William & Mary, I was really focused on how long-term exposure to mercury affected adult birds. But at Tech, I want to understand how exposure to lead, another heavy metal contaminant that's really difficult to get rid of, impacts things like inflammation and immune function, brain development, and consequently behavior and learning in adulthood. And I'll also be doing this in the zebrafinch, which is handy because I already know how to work in this system. And I think this is really important because there's a lot of foundational work on the effects of really high levels of contaminants on wildlife. However, there's a lot of different contaminants out there. You know, lead, mercury, cadmium, all of these different, I'm biased towards heavy metals because that's what I study, but they're here to stay and they're very difficult to get rid of. And I think what we're going to see a lot more of is wildlife being exposed to these survivable levels of contaminants, but over longer periods of time. So I think by understanding the effects of long-term sublethal exposure on songbirds, hopefully this can help us to better understand why these birds are declining in number and maybe even help us to, fix that. But at the same time, songbirds share a lot of their environment with us. So my hope is that by studying some of these effects in birds and understanding, the impacts to our avian neighbors, this work can also give us some insight into human health. And with that, I'd like to thank my committee at William & Mary, so Dan Crystal, Eric Bradley, and John Swaddle, as well as all of the people that worked with me on this, the Virginia Society of Ornithology, and my other funding sources. And I'll take any questions.
Great, Casey. That was a great talk, and you did a great job of really putting it into perspective of why we should care, for sure. All right, so here's a couple of questions.
Oh, sorry. Do you want me to stop the screen share?
It's up to you. Yeah, that would be good. All right. So it looks like, let's see, Nick has a question. Have you seen a difference of mercury on insectivorous birds versus birds that only eat plant material. I'm curious how mercury gets into their system if the food source could affect that.
Yeah, so sort of. This was not my work, but someone I used to work with was looking at this a bit, and we suspect that these songbirds are maybe getting it by eating insects that either the insects themselves, like spiders, consume insects that are coming from bodies of water, or the birds are eating insects directly that are, say, like hatch in bodies of water and then leave. That's sort of what we suspect might be happening. There's also the possibility that, the floodplain, so like the soil around the river or the pond or whatever body of water is being contaminated with mercury by being in close proximity to the water. And then insects from that sediment are acquiring it and the birds are eating them. But that's still an area of research, I think, that's actively being pursued, but it's not really my line of work. But that's sort of what we think might be happening. That's a good question, though. I think it's an interesting idea.
Great. All right. Thank you, Casey. One more question from Jason is asking, what's known about how methyl mercury and/or lead affects the behavior of the prey?
Oh, that's an interesting question. I don't actually know a lot about that, to be honest. That's a really good question, though. My focus is always on the birds. I think so. I haven't really thought about that too much. But I mean, I know, like, I'm trying to think. So I know in raptors, for example, they tend to get rodenticide poisoning because the rodents that are poisoned move a lot slower and are easier to catch. And high levels of lead can also have similar effects on animals. It kind of, when you are exposed to really high level of lead or mercury, it has some neurological symptoms that can make it so that animal might be a little shakier or not as responsive to their environment. The levels that we see in songbirds, you actually don't see that effect at all. You wouldn't even know they had mercury in their system, but it's totally possible that their prey items have something like that going on.
Wow. I think that's a great answer for someone who doesn't study that aspect. Great. Well, thank you so much, Casey. We'll give you a round of virtual applause.
Thank you.
And I just actually realized that I forgot to tell you about the Murray Award. So sorry, Isaac, for just a second. I'm going to just give a quick overview of the Murray Award, since Patty did a great job of telling us about the Conservation Award. So, and also just to reiterate, those of you who are members of VSO, your membership fees go directly towards supporting these students and their projects, and any extra donations. So it's really great. And I'm so glad that we're able to be here tonight to share this with such a larger audience, actually, than what we typically might have at an annual meeting. So just real quickly, the Murray Award was established in 1980 to honor Reverend J.J. Murray Sr., who was a charter member of the VSO and an editor of the journal The Raven since its inception in 1969. The award is designed to promote graduate and undergraduate research on Virginia birds, and the VSO board typically allocates between $1,000 and $3,000 a year for 1 to 3 projects to support. And the proposals are typically judged on their scientific merit and their likelihood that they will make meaningful contributions to our understanding of Virginia avifauna. And since 2000, when records really began to be kept, and that was by Gene Sattler, 52 awards totaling $50,000 have been awarded to students across universities in Virginia as well as in surrounding states. So it's really a tremendous amount of funding has gone to students, and that has led to all kinds of discoveries and lots of scientific publications and careers being launched. So it's really exciting that the VSO provides these funds. All right, so our next speaker and young ornithologist is Isaac Van Dienst. I hope I'm pronouncing your last name okay there. He received a Murray Award in 2022. He is a PhD student at Virginia Tech, also working with Kendra Sewell, where he's studying song sparrow behavior in urban and rural areas. So I'm gonna just let you share your screen and go for it.
All right. Let's see. All righty, you guys just have the presentation screen, correct? Not the. All right. Well, let's see. I'm super excited to be here today and talk to y'all about some of my dissertation work, going all the way from bugs up into birds. All right. All right. So I want to start by asking a bit of an odd question, which is if you've noticed how dirty your windshields have been getting lately. Because back in the day, you used to hit so many bugs that driving on the highway, you'd actually have visibility problems. Whereas today, you can go an entire day with only a couple splats. So this anecdote is known as the windshield phenomenon. And unfortunately, it has been vetted by multiple studies, finding that arthropods are indeed seeing dramatic declines across various habitats. So for example, like seeing 70% drops across just a decade or two in arthropod abundance. And that's some of these studies even go and look at the bird population responses as well and tend to find them declining, unfortunately. And these are done, to be clear, in their native habitats. So understanding then how arthropod communities are impacted by urbanization is crucial at a time where they're seeing these dramatic decreases in abundance and biomass across their native habitat types, where you'd expect them to be doing better than they would in an urban area. Because as urbanization increases, organisms acclimate to their surroundings with varied success, which leads to changed communities over time. And for such an important and widely influential taxa such as arthropods, this can affect songbirds and lizards and any other predators that might be relying on them. So, habitat changes affect birds too. So the Sewell Lab typically studies how urbanization might be affecting our local songbird, the song sparrows, across our urban and rural habitats. And something we've noticed over the years is that anecdotally, we appear to have much smaller territories and much higher concentrations of song sparrows in our rural compared to our urban habitat. And this could be driven by a lot of potential factors, but one that I've really focused in on is food availability. So arthropods specifically are a really incredibly nutritionally rich food item, and song sparrows, they're generalists typically, but during the breeding season, they're raising their young, they switch to about, feeding young about 50% arthropod. So it becomes a really big part of their diet. And so I set out to see what was going on the landscape from both the prey and the predator perspective. So there's a good deal of uncertainty in the literature when I first started looking at how arthropod communities are responding to urbanization. So I decided I needed to go ahead and survey my own sites to see what was available to our song sparrows. So prior work has shown that urbanization often decreases abundance, diversity, and biomass in our urban sites, but that they are fairly site dependent. And then there's also this effect of we see a loss of predatory arthropods that are the cream of the crop from a nutrition standpoint, such as spiders and carnivorous beetles. And then there's also a whole suite of new challenges that birds have to handle when they're in urban environments. But I'm mostly going to be focusing today on how urbanization affects the number, the mass, and the diversity of arthropods in the landscape, leading to the deceptively simple question, how do our urban arthropod communities differ from our rural ones? And that's going to be the bulk of the results today. But then I have about 5 minutes that I'll be moving on to talking about the bird side of things, looking at how nutritious the diet of urban and rural song sparrows are looking to be. All right. So how can we actually measure the arthropod community? Well, we can look at it in a few different ways. We have biomass, which is just the actual mass of all the arthropods being sampled. We have abundance, which is the total number of arthropods being sampled. And then we have diversity, which is just the makeup, how many different kinds of arthropods are in one area versus another. And I had a bit of a challenge because we were doing this at Tech and at rural sites. The rural sites weren't much of a problem, but I had to figure out a way to sample my arthropods without digging holes all over campus and really annoying the ground crew. And so after getting some great help from the entomology folks, I settled on vacuum sampling, which is even sillier than it sounds. So this is my little contraption. I've got it's like an electric leaf blower with a vacuum attachment. And then I've got my little paint strainer bag with a little hair tie on there. And I would walk, 50 meters, and every step I take, I swipe and, pick up the bugs, and unfortunately a lot of debris as well, which here looks fairly normal, but it's definitely a look when students are walking on the sidewalk and you are vacuuming the sidewalk. But then I would take it back to the lab and separate out the arthropods into taxonomic order, and then count them and take their mass as well. And I want to give you a quick visual of what our sites actually look like. So we have 3 urban and 3 rural sites that have been in use for about a decade, but here's just a sample of them. So here's one of our VT sites. And this yellow line here is our transect, or the walking path. And so even though you see all this, impermeable surface, all of these buildings and sidewalks / roads, you do see that this 50 meters does cover a fair bit of green space. And then like these bushes are mulch and also contain great habitat for arthropods. So it's not a barren wasteland by any means in our setting. However, when you compare that to our rural sites, it's just lush. Just to do a marsh, this is like a nice marshy riparian area. You might expect to see far more arthropods in that rural setting. And so what do we find after counting tens of thousands of bugs? Kind of what I alluded to. So for abundance, we have here habitat type on the x-axis with rural on the left and urban on the right, and then we just have total abundance on the y. And this is a stacked bar plot, which with each color representing a different order. So we have our spiders, and we have our flies, our butterflies, caterpillars, moths, our beetles, grasshoppers and crickets, bees, ants, and wasps, our true bugs, such as stink bugs and leaf hoppers, and then anything else that didn't fit into that got shoved into other. And visually, you might think this is pretty different, and that is what our models confirm, that there is a much higher level of abundance in our rural sites compared to our urban, and this held but when we looked at individual orders as well. So moving on to biomass, or how much actual mass is out there on the landscape, we see an even stronger example of the same trend, really, with rural having much higher biomass than our urban sites, and the models confirm this. There's also a couple other effects that I'm not going to get too deep into today, but there was a day of year effect where we saw increased biomass as the season went on. But the thing I do want to spend a little more time on today is this top salmon colored bar. So I've talked a fair bit about the predatory arthropods, or the protein rich, So the ones at the top, the spiders being at the top of the food web, really does make them higher in protein especially, which is obviously vital for growing birds. And I really want to draw your attention to the size of this versus the size of this. So clearly far more spiders. In general, across the board, there's far more per order as well. And then I also highlighted our lovely lepidopterans, or caterpillars, since they're such a favored item for a whole bunch of different insectivorous songbirds. All right, so moving on to diversity, we see the same thing of higher diversity in our rural habitat. So we have habitat type on the x-axis and Simpson's diversity on the y, which is just a special measure of diversity that's applied for common things, so works better for our arthropods. And so this is statistically significantly different, but there is the caveat that this is only at the taxonomic level of order, so we're not looking very precisely. This isn't looking at like genus or species. There could be changes to the significance of the effects here. Honestly, if I was going to throw a guess out, I'd say that likely we would see this an even more significant effect here. All right. So really, to summarize what I've shown you today on the arthropod side of things, our urban habitats have lower abundance, lower biomass, and lower diversity compared to our rural birds, which has some implications for our birds. Or sorry, I think I said bird, on the bug side. But so biomass availability is directly tied to predator abundance. There's a pretty big picture. Obviously, if you have more food available to you, you can do, your population will do better. But there's some really interesting nuances here. For example, 10 grams of spiders is probably worth more than 10 grams of ants. And therefore, there's some really interesting questions you can ask surrounding individual variation in development and physiology around foraging behavior and all that. But the message I want to leave for this section, and I'll come back to it, is that conserving arthropods does directly lead to conserving their predators, such as songbirds or lizards. So even if you're not a big fan of creepy crawlies, they do they keep the lights on for the birds. So what are the actual effects that we're seeing on predators? So I want to look especially at consequences in the realm of protein. So our open cup nesting birds, and I mean, open cup nesting birds in general grow quite quickly. And for our system, our birds go from, these are technically like day 3, but at day 0, they're about 2 grams. And in the course of 10 days, and if they get adequate nutrition, they end up being about 15 grams before they leave. So this is very rapid. And I decided I really wanted to focus on not just protein, but specifically amino acids, the specific building blocks of protein, because certain ones are more directly tied to, are known to be more directly tied to like muscle formation than say a protein that's used for transporting other stuff. So that's really why I wanted to look at this. A bunch of other directions you could go, but this is really the one I was excited about. All right, so what did we actually do for, how did we go out and get our samples? So we collected blood samples from our 20 rural and 40 urban nestlings. We were tracking these guys for multiple other studies, and we were taking blood samples as possible, or whenever possible. And then we looked at 36 different amino acids and their derivatives, but we're especially interested in the essential amino acids connected to growth. And as a quick reminder, essential amino acids are those that you can't make yourself. So the bird has to get it from its diet. It cannot synthesize it inside its own body. And then we use a very fancy mass spectrometry machine, which is a lifesaver. It takes a bit of time to get set up, but it finds incredibly precise concentrations of these amino acids, out of a single sample. So very good when you're taking nestling samples and getting 40 microliters on a good day. All right. So moving on to what we found. So this is going to be habitat type on the X, and then rural and green, urban and gray, and then different types of amino acids on the Y. So lysine was picked because it's essential and is directly connected to pectoralis growth, and lots of work's been done in poultry on it. And you can see it's very excitingly, there's no real difference between the two visually, but also in our models. Similar reasoning for methionine, also an essential amino acid. Looks like it might be something going on here, but nope, it's not significantly different. And then glycine is one I'm one of my favorites to look at because it's not essential in adults, but it's either semi-essential or fully essential in nestlings. More work has been done, so that's an exciting one. But once again, we're not really seeing a difference here. And then that's kind of the trend we're seeing across the board. So here's when they're all put together, total amino acid concentrations. We are seeing that there's no difference between these amino acids by habitat type, and the vast majority of other. And then breaking it down, I only showed you 3, and in the culmination, there was only a few of all those analytes that actually did significantly differ, and the ones that did differ didn't really have like a strong before the fact reasoning for why I would pick them. So interesting findings here. So consequences for our urban birds. Yeah, this is kind of where I revealed the title was a bit of a bait and switch. There's actually really not much evidence that our urban song sparrows are compromised. Despite all the results I showed you with the wildly different arthropod communities, there doesn't seem to be much of a difference in the individual nutrition for these guys, which might be surprising, but it really kind of backs up a lot of work that the Sewell Lab has done recently and in the past few years, showing that our urban song sparrows are actually doing great. They're outcompeting the rural in many ways and seem to be fine. The huge caveat, though, is that this is only on the individual level of scale, and that if we look at population level effects, they're likely to be we might have different findings. I would assume there would be a much lower carrying capacity in our urban habitat than our rural. And I just want to leave you with the call to action of save our bugs to save our birds. There's a lot of work that's been done out there to show how to do this, which is awesome. And then I'd like to finish by thanking my co-authors, committee, and lab members for all their invaluable feedback, my funding, the VSO Interfaces of Global Change, NSF, and SICB as well. And with that, I'm happy to take any questions.
Awesome, great. It's very interesting research. It makes me thinking about the higher abundance of birds in the rural areas, like they're kind of figuring it out, like they're like where there's more food, there's able to be more birds, and really that idea of sometimes, we can measure density and that can be an indicator of habitat quality. Sometimes it can't, but sometimes it can. So that's kind of a cool result there. So a couple questions. So Nick is asking, do you think that they're eating more to make up for a lack of nutrition? I'm guessing in the urban areas.
Yeah, so this is I was right on the time, like pushing the timeline with just these bits. So my other chapter that I've been working on is looking at stomach contents and nestlings. So I do actually have an answer to this, which is yes and no. So on the abundance side of things, urban birds are actually eating more, but when you look at total stomach mass, content mass, and then if you look at like invertebrate content mass within the stomachs, there's no significant difference between the habitat types. But kind of my thought process is like, well, looks like urban birds are managing to compensate, even if they have a lot fewer things available to them. It still doesn't seem to be problematic for them.
Okay, great. Diane asks, in defining rural, how did you consider and separate out agricultural areas where unfortunately farming trends have negatively affected insect diversity and abundance?
Yeah, so this is definitely a bit of a effective, like the way we just picked our field sites years like before I got to the lab. So they were picked based on percent like land cover, essentially like impermeable surface. So our rural sites, if you're familiar at all with the Blacksburg area, we have a really a riparian restoration area that is incredibly rich. Then we do have an experimental farm. Kentland Farm is one of our sites, and then we have a public park. Those are our three rural sites. So we do have a bit of a mix there, and there are site differences. With the farm being the if I recall correctly, being the lowest for like biomass and abundance. Yeah.
Actually, I have a question kind of related to that.
Yeah.
When you were using the kind of reverse vacuum to sample your insects, did you use that same method across all of your, so like even if like you were
Yeah.
so it was always the grassy areas that you were measuring the ground?
Yeah. So there were, so basically, because we obviously hit like bushes and everything, and there the rough guideline was keeping the tip pressed to the substrate, whether that substrate was a grass where it was nice and easy, or a bush where it was a little more difficult, but extending it through and trying to control for the amount of time I spent on the transect and all that. So it wasn't just flat grassy areas. It was
that makes sense for lepidopterans and such.
Yeah.
Okay, and then Chris asks, did you sample any urban areas that were specifically planted as pollinator habitat?
So not to my knowledge. There's a couple places that are really wonderful at Tech that would be good for that. Something I didn't mention, because would take too long in all the methods, all the transects were derived from, so these were done in 2020 and 2021, but all of the transect start points were 2019 song-sparrow nests. So they were placed in a way that should have some relevance to foraging, but I did not try and bias them in any way. So I don't believe off the top of my head, because there's only 4 transects per site, run multiple times, but no, I don't think I got anything that was specifically pollinator focused.
Okay. Any other questions? Don't think so. Okay, well done, Isaac. Thank you. All right, and I guess we are kind of moving through this a little bit more quickly than I expected. So that's good that we won't keep everybody so late in the evening. But we do have one final speaker that I actually got a prelude of this at the AOS meeting. So I'm excited to see this again because it's really exciting stuff. So Garrett Rhyne received actually 2 Murray Awards, one in 2021 and one in 2022 for his master's research at Louisiana State University. Although he started out as an undergraduate at Virginia Tech, working with Swainson's warblers, but he's working with Phil Stouffer to study Swainson's warbler migration. He plans to graduate this December and then continue his career in avian ecology and conservation. All right, Garrett, it's all you.
Awesome. Sorry, my computer's messing up, so my face isn't going to be matching up here with my
And actually,
screen.
you know what, Garrett, we're actually seeing, we're not seeing your presentation. You're seeing your presentation, but it's in the presenter mode, not the,
Oh.
you know.
Hold on one second. How about now?
Yes, that looks great.
Okay, perfect. All right. Well, good evening, everyone. Thank you very much for having me. My name is Garrett Rhyne, and I'm a graduate student at Louisiana State University, and I'm really happy to be here tonight to talk to you all about my research with Swainson's warbler migration ecology and connectivity. So when we're studying neotropical migrants, a lot of times we have big emphasis on the breeding sites, which is very important. But as we know, a lot of these migrants only spend about a third of their life annual cycle on the breeding site. So in order to properly conserve them, it's really important for us to have to be able to track them throughout their annual cycle, to know where they are spending time in their fall and spring migration, and especially where they are overwintering. And not only that, but it's also really important for us to be able to connect our breeding and wintering populations, which is known as migratory connectivity, so that we know where each population is going for the winter and to know if they're staying together or staying separate, which has huge implications for conservation. And additionally, it's important for us to know when these birds are migrating, because especially as a warming climate is changing the phenology of resources available for them as they migrate, this could threaten their ability to migrate properly. And so one species that would greatly benefit from this full annual cycle evaluation is the Swainson's warbler. Swainson's warblers are these charismatic ground-dwelling species of conservation concern that inhabit extremely dense forest understories all throughout the southeastern United States. And their range is broken up into three primary breeding subpopulations, including the Mississippi Alluvial Valley, the coastal plains, and the Appalachian, which is the least studied and most poorly understood. And generally, we know that these birds are wintering throughout the Yucatan Peninsula and the greater Antilles, but there's never been any tracking done of them to be able to connect those breeding sites to know where those birds are going for the winter, and also the full extent of their wintering range. And so for my master's research, I wanted to fill in this really important information gap by tracking Swainson's warblers throughout their annual cycle to determine those wintering locations, to then evaluate that degree of connectivity between the breeding and the wintering locations, and then also to be able to characterize their migratory phenology and their behavior. So in order to do that, sorry about that. In order to do that, I gathered up a collaboration of wonderful researchers from across 7 different states, including 2 wonderful researchers and mentors of mine, including Dr. Ashley Peel at Virginia Tech and Dr. Leslie Bullock at VCU, in order to tackle this range-wide project. And we're lucky enough here in Virginia to have both the coastal plains population and the Appalachian population. And if you're familiar with those habitats of Swainson's warblers in Virginia, you know that they're quite different across the west and eastern side of the states, where in the Appalachians, they occupy these extremely dense rhododendron and mountain laurel thickets and very steep hillsides. And then in the Great Dismal Swamp, these birds are occupying just absolute wall thickets of cane and greenbrier throughout swampy bottomland hardwood forests. And so we chose these different sites, and with the wonderful work of both my research collaborators as well as some technicians, including Nicola Novak at VCU and Hannah Waite, who was a research technician, we went out to these field sites and we went and captured male territorial Swainson's warblers on their breeding grounds. And we used audio playback, caught them with mist nets, put bands on them, got a lot of measurements. And on each one, we attached a leg loop harness with their tracking device. Now, when we're deciding on how to track a small bird, and I mean under 25 grams, it's a little bit more complicated than being able to put a big GPS tag like with that wonderful data that we saw with the Kestrel, right? It's a little bit more complicated because these birds are so small and they can't carry a heavy tag. So one option for small birds is an archival GPS tag, which you have to retrieve, which is very accurate. But again, this kind of GPS tag is unfortunately too heavy for a Swainson's warbler, not to mention they're extremely expensive. So that wasn't a viable option for us. There is a wonderful network of automated radio telemetry towers. This is known as the MODIS network, which is greatly expanding, especially in the state of Virginia, which as a tagged bird flies nearby one of these towers, it's going to go to a central database so we can track their migration. Unfortunately, there isn't a good enough network in the wintering locations that these birds are expected to go for this to be a viable option. So instead, we used what's called archival light level geolocators. And what these do is they record light on the backs of the birds. And from that data, we can get sunrise and sunset from every day from when it's attached to the bird, and we can estimate longitude and latitude. However, this means that our estimates are going to be a lot broader, but we're able to deploy these anywhere we need to. We don't need extra infrastructure, and they're very affordable. Now, as you can guess from the title of my talk, not only did we get to use regular light level geolocators, but we also got to deploy these brand new barometric geolocators that record atmospheric pressure as well as light. And as we would come to find out, this would be a game changer in not only our ability to track the birds, but also to study their migration ecology and behavior. And so we deployed 86 geolocators in 2021. And in 2022, we went back and recovered a whopping 36%, which for geolocators, for those not familiar, is a very good return rate. We're able to have, in Virginia, we recaptured 9 in the Appalachians and 3 in the Great Dismal Swamp. Unfortunately, when the tags were being sent from the Great Dismal Swamp for data download here in Louisiana, the package was actually stolen in transit, very unfortunately, and so we lost that data. However, we still got a lot of really valuable data from the rest of our field sites. So when you think about a Swainson's warbler, they're crawling on the ground most of the time, and they're inhabiting this extremely dense understory. So when we're analyzing raw light data to figure out where they're located, you can imagine that there's a lot of sources of error, and you'd be right. So here's our standard procedure for analyzing light data. And on that far right map, you can see this is a bird from Louisiana. We get a likelihood map as a heat map that shows how broad those estimates are. And this for a Swainson's warbler was good quality light data, whereas bad light quality data, where it's very, very shaded, especially during the winter, just ends up with extremely broad estimates that are most likely inaccurate. And the only really important information we can get from it is that longitudinal information. This just goes to show that, using light level data, especially for a species like this, is not very accurate and not very good. And we also don't get a lot of migration information out of it either. But luckily for us, like I said, we got to try out for half of the tags that we deployed. We got to have data that records atmospheric pressure as well as light. And there is a brand new analysis called GeoPressure R that allows us to take our recorded pressure data and compare it to a weather reanalysis data set, as well as match it to the elevation based on that altitude from recorded pressure to better figure out where those birds are located. When we are looking at just this raw pressure data in terms of altitude, there's a few things that we already know that we would never otherwise know with other tracking technologies. So we know exactly when birds are at what we'll call a stationary period. So that means at the breeding site or at a stopover site. And from there, we know exactly when birds are migrating. So here, this is across a couple of days in September, we see this big spike in altitude, and that corresponds to a bird conducting its nocturnal migratory flight, which they fly at very high altitudes. And so we're able to delineate exactly when they depart, exactly when they arrive, We know exactly how long each of these flight bouts are, and we also will know how long they're stopping over for. Additionally, we know a lot of other interesting information, as this is bi-hourly data. We know the altitudes at which they're flying as well, which can be really important. And so we take those stationary periods, and we're going to basically compare it to this weather reanalysis data set. It's a fancy mismatch likelihood analysis that incorporates not only where the bird is spatially, but also temporally. And we also incorporate what's called a movement model. And all of that will eventually turn into these wonderful maps that are accurate and, very importantly, continuous location data throughout the year. Emphasis on continuous, because most of those other technologies do not continuously know where the bird is. Whereas here we know exactly where they're stopping over with a little bit more broad estimates. And so these are 3 different tracks from 3 different individuals from the Appalachians, from Breaks Interstate Park in Virginia. And they all, as you can see, utilize peninsular Florida to migrate down in the fall. And they all wintered in western Cuba, and then flew back across the Gulf of Mexico on the eastern side, and again used Florida to make their way back. So this is really important full migratory trajectories that very few songbirds have this sort of data. And the wintering locations themselves, so those long periods of time from basically October until March that they're staying, those location estimates are far more accurate than can be achieved with other technologies, down to basically anywhere between 10 to about 40 kilometer estimates. And so here is from all of my birds, which also includes tags from Louisiana and Missouri, where Western breeders, like here in Louisiana, wintered in the Yucatan Peninsula in Mexico, which was distinctly different from all the Eastern breeders, including the Appalachian birds, as well as the coastal plains, like birds from South Carolina. They all wintered in different parts of Cuba. And if you can see, these likelihood estimates are not blown up in a radial pattern like those light estimates. They're in these really weird shapes, and that's because it's following elevational gradients. So we know whether that bird is occupying montane regions or if they're down in a bottomland swamp, which can be really useful for us determining where this bird is overwintering and where to prioritize conservation efforts. As I mentioned, we're able to estimate stopover sites even for less than a day long, which is really important. And one very important value of this is we found that basically every single bird from the eastern populations stopped over in the Okefenokee Swamp region, as you can see here on the Florida-Georgia line, for anywhere from 3 to 10 days in spring migration, which means they're using this site not just as a quick pit stop, but a long-term stopover site used for refueling. And this can be extremely important for conservation efforts on knowing where best to conserve land for migratory stopover habitat. Some other really interesting information that we can gather from this sort of fine detailed data is we can plot out exactly when birds are migrating. So this is what we refer to as the phenology of migration. Here I've plotted every single bird from Louisiana to Virginia, and we can see how they compare across populations of when they're migrating. On the left side, we can see fall migration, which is very short and synchronized, versus spring migration, which for almost every bird was more drawn out and spent more time on stopover sites. And this is actually antithetical to our current understanding of how we think birds migrate. We think that birds are going to be much quicker in the spring because they want to return to their breeding territories, and these are all male birds. Whereas here we're seeing, birds are migrating much faster in the fall. So this could be, from a number of reasons, whether it be they do actually care about those winter territories, which are also important for gaining resources for surviving throughout the annual cycle. But it is also probably a product of favorable environmental conditions. So this is only 1 year's worth of data. So this might have only happened this year, and it would be important for us to track them throughout multiple years to know if this is a consistent pattern or not. And lastly, we are also able to gather this really interesting flight behavior data that has always been extremely difficult to track. These birds are migrating at night, and so our understanding of how they migrate and how high they're migrating is extremely, a lot of times intangible. And so this allows us to actually visualize these nocturnal flights really well and begin to parse out sort of the drivers behind the mechanisms that drive these relationships. So here's an example where I just plotted the time of day, or the time of after sunset, of when the birds departed that night versus how far they flew throughout that nocturnal flight. And what we found is that birds that are going very short flights, say just hopping over, anywhere from 20 to 50 kilometers, they're leaving anywhere between right after sunset to 4 to 5 hours after sunset. Whereas birds that need to make those really long journeys, say they're crossing the Gulf of Mexico, which almost all of our birds in Louisiana did, they're leaving basically right after sunset. So there is this relationship to when is the prime time to leave to maximize their nocturnal flight. And so this is just one example of the many things we can kind of glean from this atmospheric pressure data. And so in conclusions, by tracking Swainson's warblers across their breeding range, we were able to find that they do exhibit moderate amounts of migratory connectivity, which means eastern and western birds are staying separate during the wintering period, which is extremely important for us to know. But there is still some overlap in the Appalachian and coastal plains populations that winter in Cuba. It's also important for us to note that even though we didn't get data from the Great Dismal Swamp, based on this longitudinal relationship, we expect that a lot of these birds are going to be wintering in eastern Cuba and Jamaica. We didn't have any other birds wintering in Jamaica, and that's a big wintering location that a lot of research has come out of. And so we expect that that's most likely where they're wintering and following this longitudinal relationship as well. And so we've been able to identify not only important wintering sites, but important stopover pathways as well. And what may be even more importantly, and is something that will be coming out, will be published soon in Ornithology, hopefully, is that we got to basically be the guinea pigs of using these barometric geolocators on small migratory birds. And it allowed us to gather way more migratory information than we could have possibly asked for. These barometric geolocators, they're more accurate than most other tracking technologies. They have a lot of data, and they're also very affordable. So they're the same price as geolocators as well. And so this technology has the possibility of revolutionizing the way we study migration ecology in migratory birds. With that, I'm very thankful for the VSO. It's actually the VSO who I was able to get funding for as an undergraduate at Virginia Tech with Dr. Ashley Peel when I studied Swainson's warblers in southwestern Virginia for an undergraduate project that basically led me here. And so I am forever grateful for VSO for providing funding and support for this project, as well as many other partners, especially from Virginia. And with that, I'd be happy to take any questions.
Okay, excellent talk, Garrett. And that picture next to the...
I know, it's a wonderful picture next to the rhododendron, their favorite.
Yeah, that's great. All right, so Joe says, great talk, Garrett, and fun technology for sure. Tragic about the package theft. But his question is, seems like there's a lot of elevation change. There isn't a lot of elevation change across Florida. How does the barometric pressure data help increase the precision of those location estimates in a place like that? For example, identifying the Okefenokee.
Yeah, great question. Yeah, a lot more weight is carried by that spatial temporal variation in atmospheric pressure. So this is hourly data that's being compared to. So unlike with light level geolocators where you only have 2 data points for a single day, you have 24 data points of comparing these fluctuations in pressure across the day. And it's surprisingly very accurate, even in very flat landscapes, and oftentimes can be more beneficial that they're on flat landscapes. Like when I was estimating just breeding locations to test the accuracy of birds in the mountains, if they're moving up and down in altitude, just going up and down hillsides, it can interfere with our ability to estimate. So actually being on flat land can actually benefit us. And I will say that again, the estimates for migratory stopover sites of, even a day or two is going to be broader than our wintering estimates, simply because it's far less data. But we're still able to get at the general region pretty well, just from that spatial temporal variation. Oh, you're muted, Leslie.
Thanks. All right, so that's a good answer. So let's see. Jason asks, out of curiosity, could you look for a correlation between these few birds' migration flight and stopover dates and Doppler radar-based big migration nights?
Yes, yeah, absolutely. This is actually something that could easily, using this sort of data, can help ground truth a lot of that weather radar data as well. And I think it's also important to mention that in this migration data, so in the movement model from going from one stationary period to the next, wind speed and basically local weather environments, which is pretty fine scale at a 0.25 degree grid, is actually incorporated into those movement models. And so wind direction and the tailwinds from that night are incorporated into the direction that these birds are migrating in terms of how we estimate those locations as well. So yeah, a mix of a lot of weather and both, radar ecology that we can ground truth with to better understand, how are these birds taking advantage of weather patterns to best migrate.
Awesome. All right, Dan also asks, is the barometric sensor sensitive enough to capture weather fronts at stable latitude? If so, can it be used to contextualize migratory decisions of individuals, or is it that better accomplished with weather data from regional climate monitoring?
Yes, no, yeah, it does. So because, weather doesn't interfere with it, with our estimation, because this is a weather reanalysis data set. So if you have big weather events, it actually shows up better for those estimates, and we can learn a lot more from it. So for example, 3 of my birds in Louisiana here, we lived through Hurricane Ida in 2021. And that shows up in the data where it's this huge low pressure system, this big curve, and it matches perfectly with the location that we're at. So that I was actually able to figure out that those birds were displaced probably no more than 20 or 30 kilometers away. So it was pretty close, but they were, at a different location afterwards, because we were able to recognize that weather pattern. So yes, I think you can look at it's best to look at compared to local weather radar data in order to understand exactly what, because this pressure data isn't going to tell you what it is unless it's as big as a hurricane. But yes, short answer, yes, you can use this data to kind of figure out how it changed the migration of that bird, and maybe a little bit of decision making as well.
Yeah, it's truly amazing. I'm expecting it's Low Tech that was producing these.
Migrate Tech.
Migrate Tech. I'm expecting them to be needing to ramp up their production after this paper is published.
Yes.
So really exciting stuff. All right. Well, I'm going to pass it back over to Diane or Dixie. And thank you guys so much for all of these presentations. Fantastic.
Yeah.
Leslie, thank you. Dixie, can I jump in?
Yes, please do.
This was absolutely fabulous. All of you did an unbelievable job. I'm blown away. I'm really deeply moved too at to see how far we've come with the research that's available, with the enthusiasm from professionals and young folks like you. This rocks, totally rocks. And then we've got a couple of really, I've been seriously birding since about 2008. We have people on board here now that have been around since the 70s. I mean, Bill Williams, Bob Ake, I saw you guys were on the call. I'd love to hear how you've seen things change as well. But this really, thank you guys so much. And I'm glad we're recording it, because I, for one, will have to go back and watch this again. So, yeah, thanks a billion. And Garrett, I remember you from Mountain Lake at a VSO meeting around Atlas time, and had a blast with you and your buddy Josh Ward picking up rocks and looking for salamanders too. So glad you're doing well and sticking with ornithology. So
Great, thank you. I'd like to second that, all of that. You know, it's been amazing to see what more can be done because the technology is coming along. And what Garrett just showed us about a new technology is just amazing. I guess
I want to say one thing real quick, and that is
Yeah.
just if anyone has any recommendations for how this went, if you know, I think we would probably like to do this again in the future, maybe having you know, award and grant recipients present. So any feedback you can provide, please send me or Dixie or Diane an email and let us know.
Yeah. And if you're in doubt about an email, you can use the email that you received that had the link on it. That goes to a VSO address. So I don't know if Bill Williams or Bob Ake would like to respond to Diane's question. You can unmute yourself and make a comment if you'd like.
not to put anybody on the spot, but I can't help but think, you know, having been around that long, maybe go ahead.
I'm kind of in the dark here because I got the lights out in the back, but yeah, all of this is just absolutely fantastic. I look forward to the presentations that we have for our William and Mary students every year. Casey was part of all that. And these are the kinds of things that give you a great amount of hope for what young people are looking at and how they're approaching their disciplines and the things that we're learning from them. A theme that came out across all of this is how much interconnection there is. When we went from weather to different kinds of insects throughout this talk, and there's so much more depth to this than many people tend to realize. It's not just birding. It's a fascinating use of science to improve everything that we're doing to look at it. And I've got to say, Leslie, absolutely fantastic the way you pulled all this together. And you've been after us to make this happen, and it worked. Way to go.
Well, I won't add too much to that. I do go back centuries. And one of the best things about the VSO annual meeting was always the paper sessions, and I always attended them. They were really great, and that's why when we had the one down at the swamp a couple of years ago, when unfortunately, Garrett, we didn't do your runs, but I guess it wouldn't have mattered anyway. It would have been lost in the mail. But anyway. I think it's great, and I think that what you're seeing is the continued enthusiasm in doing this, and what impact some of these new techniques have at settling questions that seemed unanswerable years ago. So it's really good to see all this progress being made, and I always knew those Swainsons were headed down to Cuba, and it's good to know. Anyway, it's great to see.
Thank you. I guess one thing I noticed too in the slides folks had about who their funders were, there were several state ornithological societies mentioned in addition to Virginia. So I guess that what that shows is that we have these state organizations that have a commitment to supporting science, and that's certainly strong with the VSO. So
yeah.
I guess if there are no other comments that anybody wants to make or questions, we can call it a night and call it a very successful night. I'm really pleased so many participated and with the quality of the presentations that we had. Looking forward to the next round of grants.
Thank you, everybody. This was
Thanks
fabulous.
very much, Patty, Leslie, everybody. Thanks a lot for coordinating.
Thank you.